Hello and welcome. Let's talk today about muscles and muscle tissue. When we start off, we'll talk about some functions and we'll talk about skeletal muscle, and then we'll talk a little bit about smooth, and then we'll check out cardiac when we get to the chapter in relation to cardiac muscle. When we talk functions, first function is producing movement. Now, when we talk skeletal muscle is going to be responsible for producing local motion, right? Moving from place to place, and manipulation of our environment. So it allows us to move from place to place and manipulate our environment. Now, when we talk about cardiac muscle is going to be responsible for moving the heart, which then basically moves blood. When we talk smooth muscle and movement, smooth muscle, it courses blood and food stuff, to just name basic things, throughout the body. Let's talk then maintaining posture, stability, basically here. Maintaining posture. Now here we're talking mostly skeletal muscle, right? Skeletal muscle is going to be continuously making tiny adjustments, tiny movements to counteract gravity. Stabilizing our joints. When we talked about our joints, we said our joints were the weakest part of our skeleton. Nonetheless, they're able to resist various forces that are going to malalign them, right? And that's because of our muscles. So when the muscles contract, basically this is going to cause the joints to be strengthened. It stabilizes and strengthens the joints. So when our skeletal muscle contracts, this stabilizes and strengthens our joints. Generating heat. We have another function here, generating heat. Muscles generate heat as they contract, and heat maintains at normal body temperature. Right? For example, shivering, we have muscle contraction. There's some additional functions I'd like you to know about. Here is, again, skeletal muscle, protecting our internal organs, protecting our viscera. Then we talk smooth muscle. Smooth muscle forms valves that are going to be responsible for regulating the passage of substances. It also dilates and constricts our pupils. And then forming the piloerector or erector pili muscle, the goosebump muscle we saw there also. Next, then let's talk a little bit about some properties, some special characteristics of muscle tissue. Now, when we talk about some special characteristics of muscle tissue, some properties of muscle tissue, the first characteristic I'd like you to know about is excitability. It's responsiveness. Basically, muscle tissue has the ability to receive and respond to a stimulus. It has the ability to receive and respond to a stimulus. Second is contractility. Muscle tissue has the ability to shorten forcibly when adequately stimulated. It has the ability to shorten forcibly when adequately stimulated. Conductivity, we can see another one. We have another one. That's the ability to spread electrical impulses through its tissue. It has the ability to spread electrical impulses. Extensibility, we have another one. Extensibility, meaning it has the ability to be stretched or extended. Muscle tissue has the ability to be stretched or extended. And then elasticity. Muscle tissue has the ability to recoil, and resume its resting length after having been stretched. So you see, these are all properties, some special characteristics in relation to muscle tissue. Right, we didn't talk about these characteristics in relation to epithelial tissue. Okay, it had its own. Connective tissue, it had its own. Right, nervous tissue we saw, it had its own. So these are of muscle. Next, then, let's move down. Let's talk about the different types of muscle tissue. Now, when we talk about the different types of muscle tissue, we have seen and we've visited and we've checked this out before. We have three types of muscle tissue. When we talk muscle tissue, we have skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. Now, here we can see we've got all three types right inside of here. We'll go through and we'll talk about all three types, and we'll check each of them out and check out some characteristics in relation to each of them as well. And then, like I said, we'll go through each of them individually as we progress through chapters throughout the semester. Now, when we go through and we talk skeletal muscle, you can see, is made up of skeletal muscle tissue, which is made up of skeletal muscle cells. So when we talk skeletal muscle tissue is striated. It's voluntary. It's under voluntary control. And it is responsible for the overall body mobility. Skeletal tissue is found attached to and covering our bony skeleton, and then sometimes to the connective tissue there as well, as we mentioned. Now, when we talk appearance, let's break this tissue down to one muscle cell. Now, when we look at one muscle cell, you can see these cells are going to be long, cylindrical shaped cells. They are the longest muscle cells that we have. And they have these obvious striations, as we can see. These obvious stripes, striations, the term we'll use. Multinucleate cells. Multinucleate cells. And we'll look at all of these striations under the microscope in greater detail as the class progresses today. Next in here, we can see we've got cardiac muscle cells. Cardiac muscle cells, which came from cardiac muscle tissue, right, which is found making up the walls of the heart, basically. Now, when we go through and we talk cardiac muscle tissue, it's located only in the heart, which is the body's blood pump. The appearance, striated. You can see that there as well. All these striations, just like you can see in here. Now we talk appearance, striated. Now these cells, we said they are uninucleate or binucleate, generally uninucleate. And they are branching cells. They interdigitate, right, with the next cell. We said at the, very good, intercalated discs. I told you not to confuse them with the nuclei. Now, when we go through and we talk smooth muscle, third type of muscle tissue, smooth muscle. Smooth muscle is going to be found located in the walls of our hollow visceral organs, such as the stomach, the urinary bladder, right, respiratory passageways. They're all going to contain smooth muscle. Now, when we talk about one smooth muscle cells are smooth, hence the name, so they have no striations. Smooth spindle-shaped cells, uninucleate cells, centrally located nucleus, spindle-shaped, fusiform. So fusiform, spindle-shaped, uninucleate cells with no striations. With no striations. Next, let's move to skeletal muscle then in greater detail. So let's talk more about skeletal muscle then in greater detail. Now, when we talk skeletal muscle in greater detail, here we are able to appreciate, now, basically one component in relation to skeletal muscle we'll go through and we'll check out. First thing I'd like you to know about is that skeletal muscle is considered an organ. It's considered an organ, so we are revisiting that definition there. So it's something you want to reiterate and make sure you have down. Now, when we talk about skeletal muscle being an organ, nerve supply it has, right? When we talk about nervous tissue, we're going to talk epithelial tissue. We're going to talk about obviously muscle tissue there, and then connective tissue. So 4 primary tissue types. So here when we go through, we talk muscle as an organ. So muscle is made up of several tissue types. Skeletal muscle fibers obviously dominate, but blood vessels, nerve fibers, connective tissue are also present. So let's talk first nerve supply. Nerve supply, nerves you're going to see, they're going to be found. They'll be entering and exiting from the central part of the muscle. Now when they enter, they're going to branch profusely. They're going to branch profusely through the connective tissue sheets to make their way into, innervating then, our muscle fibers. Blood supply, muscle tissue has a rich blood supply. It has a rich blood supply because the contracting muscle fibers are going to need continuous oxygen and nutrients. They can also have one or more vein as well, helping to remove waste so nothing builds up. Very nice. They have also what we call an epimysium to them, as we can see here. So this is going to be the connective tissue component. So we've got muscle tissue, nervous tissue, blood supply we mentioned there, because epithelial tissue. And then here we've got connective tissue. So when we talk connective tissue, we have the epimysium. The epimysium is the overcoat of the muscle. It's the overcoat. And it's made up of dense, irregular connective tissue. Dense, irregular connective tissue that is found surrounding the whole muscle. That is found surrounding the whole muscle. Now, here in relation to this epimysium, let's talk attachments. Basically, how this muscle comes into contact with the bone. So when we talk attachments, here you'll see muscle is going to come into contact with the bone a couple of different ways, thanks to the connective tissue. So first we have what we call direct attachments, and second we have indirect attachments. Now when we talk about direct attachments, okay, so when we talk about direct attachments, I won't say indirect attachments because then you're going to think I'm talking indirect attachments. So when we talk about direct attachments, here we have the epimysium of the muscle fusing directly into the periosteum or into the perichondrium. Now, when we talk about indirect attachments, here, muscle's connective tissue wrapping, this epimysium, is going to extend beyond the muscle. It'll extend beyond the muscle, either as a rope-like tendon or a sheet, or a sheet-like aponeuroses. A broad sheet of tendon, aponeuroses, which then connects to the bone. So here we could see a broad rope-like tendon being formed. So this is an indirect attachment. Next, then let's move further into this muscle. So when we look at this muscle in greater detail, we can see this muscle is made up of all of these fascicles. Now let's pull out one fascicle. Here when we pull out one fascicle, this is what we see here. These fascicles are groups of muscle fibers within skeletal muscle. And they resemble basically bundles of sticks, as we see here. Like they have a whole bunch of bundles of sticks. Now, in relation to the fascicle, we have what we call perimycium. So these fascicles are wrapped in perimycium. This perimycium is a layer of fibrous connective tissue that's found surrounding each of the fascicles. So we can identify it here, and we can also identify it right inside of here, in between these fascicles. Let's move even further then into the muscle. We move further into the muscle then, we've got our muscle fibers. So each fascicle, you can see, is made up of a whole bunch of muscle fibers. Now here what we're going to do is we're going to pull out one of those muscle fibers and we're going to examine that. Now one muscle fiber is a long cylindrical cell. So we could say muscle fiber or muscle cell with multiple nuclei as we've described. So let's look at that muscle fiber then in greater detail. So let's move a few slides. Here is where we were at. We moved down to the fascicle, and we had the fiber. So we're pulling out this fiber, and that's what we're looking at now in greater detail. We see here. Now here we can see in relation to the muscle fiber, we have what we call endomycium. Endomycium is a sheath of fine areolar connective tissue found surrounding each individual muscle fiber. Beneath that, you can appreciate the sarcolemma. That's the plasma membrane. It's found beneath that endomycium. It's basically the cell membrane. Nuclei, we said, multiple oval nuclei. That's why this muscle is considered multinucleate. And then here we can also appreciate, you can see mitochondria. Let's zoom in now on this image here. When we zoom in on this image here, that's what we're seeing there. So a whole bunch of mitochondria you can see right inside of there as well. Now, here you can see the endomycium has been removed. Now all we have is the sarcolemma. So this is the cell membrane. Now inside the cell we have cytoplasm. So here we call it sarcoplasm. Sarcoplasm is the cytoplasm of the muscle cell. Inside of the cytoplasm, we're going to have glycosomes. Glycosomes are granules of stored glycogen, if you recall from biology. And what glycogen does, it provides glucose during periods of activity. It provides glucose during periods of activity. We also have in here myoglobin. Myoglobin is a red pigment that stores oxygen. In addition to the usual organelles, a muscle cell contains three structures that are highly modified. Number one, you can appreciate in here, myofibrils. Number two, we're going to go through and we're going to check out the sarcoplasmic reticulum, aka in a normal cell, endoplasmic reticulum you learned about, and then T tubules. The third being T tubules. And we'll look at all of them as we progress through here. So here, actually, we can jump a bit. Right in there, you can appreciate the sarcoplasmic reticulum. Yeah, we will be talking about this, so I don't want to jump the gun. Next then, let's talk about the myofibrils. So let's look at these myofibrils in greater detail. So this is what we have. This is where we were at. We're going to get this myofibril, and we're going to throw the sucker under the microscope, basically. Look at it in full-on detail. So here we can see then, this is what we have. This is what it looks like. Coming back, you see that there? And this is what we have. These myofibrils, they are rod-like elements that come together to form a muscle fiber. Rod-like elements that come together to form a muscle fiber, form a muscle cell. They are the contractile units. Right, the nucleus doesn't contract. Right, what'd you learn about the nucleus? You should know all that from bio. The mitochondria, does that contract? No, I think I remember from like elementary, it's the powerhouse of the cell. So no contraction there, no. Myofibrils. So they're the contractile units. Now, when we talk about these striations, you see there, these striations, they're repeating series of dark and light bands. Or you can say A for dark and I for light. Repeating series of A and I bands. A and I, A and I, A and I, or dark and light bands, dark band, light band, dark band, light band, dark band, light band, dark band, light band. Okay? So they're repeating series of dark bands and light bands, A bands and I bands. Let's look at then these A bands and I bands and all of that in greater detail. Right? I want to know what that looks like. So that's what we're seeing right inside of here. So first here we can appreciate the A band. Let's check that out. So here we have the A band. It runs from here, this side here, all the way to about right inside of here. Now this A band, you can see has, in relation to it, the H zone. Like let's say, for example, construction zone from Bear Valley to Nisqually exit. Okay? So from here to here, so it's a zone. When we talk H zone, the H zone is this lighter mid-region. It's this lighter mid-region. It's the lighter mid-region where the filaments are not going to significantly overlap. Now, when we go through and we talk about this H zone, the H in H zone stands for basically heller, the brighter in German is what it stands for. And then the A band is the anisotropic band. So here then we've got the H zone. So the H zone, the heller zone, it's also known as we said, is the lighter mid-region. It's this lighter mid-region where the filaments don't overlap. Where the filaments don't overlap. Next then we have the M line. The M line is basically this line of protein called myomycin, but the M line, again, it's German for Mittelscheibe. I'm not German, excuse my pronunciation and I guess the butchering of it. But M line, it's basically means the disc in the middle of the sarcomere. So here we can see that M line. Here for us, we'll keep it as a line of protein myomycin. And what it does, it holds the adjacent thick filaments together. It helps to hold the adjacent thick filaments together. Next then we have the I band. When we talk about the I band, the isotropic band, we can appreciate here now, is basically going to contain Z discs, Z lines, Z bodies, whatever you want to refer to them as. And it also stands for Zwischenscheib, I believe, Mittelscheib and Zwischenscheib. So Z disc, Zwischenscheib, and M line, Mittelscheib. But anyways, so the Z disc is a coin-like sheet of proteins. It's a coin-like sheet of proteins, and it anchors the thin filaments. It anchors the thin filaments, and it connects the myofibrils. Now, when we go through and we talk this term sarcomeres I've used here. The term sarcomeres is a term we're going to use to refer to the smallest contractile unit of a muscle fiber, the functional unit, basically, of our muscle fiber. Now, when we talk sarcomere, it's the area between two Z discs. It's the area in between two Z discs. Now, when we go through and we talk, so the area in between two Z discs, you can see from here to here, this is our sarcomere. And here we can see that's what we've magnified, and we're looking at is just one sarcomere. So again, it's the area between two Z discs. It is composed of thick and thin filaments, and it's made of contractile proteins. It's made of contractile proteins. We're going to go through and we're going to check them out. Careful examination of a myofibril shows it arises from an orderly arrangement of smaller contractile structures called myofilaments. Myofilaments, actin and myosin. So let's look at actin and myosin. Actin and myosin, they play a role in motility and shape changes. So here, first what we'll do is we'll look at our thick filaments. Now here we can appreciate the thick filaments right inside of here. And then surrounding them are the thin filaments in blue. Thin filaments, thin filaments. And then between two thin filaments, you have a thick filament. So here, when we talk thick filaments, the thick filaments are going to consist of protein myosin. Thick filaments, they extend the entire length of the A band. They extend the entire length of the A band. So here you have the H zone, the M line, we've talked about, right? So let's talk myosin. Myosin is a protein. So let's magnify this protein image here. So here we can see the thick and the thin filaments. Thick filaments, and then the thin filaments. And you can see them in greater detail here. So let's focus on the thick filaments. And what we'll do is we'll come over to this picture here. Now when we talk about myosin, myosin is going to assist in movement. Myosin, you can see, contains a tail, basically, component and a head component. And when we talk about the tail is two woven, heavy polypeptide chains. The heads, they contain two smaller, light polypeptide chains that are going to act as cross bridges during contraction. And here we could see at the level of the heads, you've got first your actin binding sites, so binding sites for actin. And also here near the flexible hinge region is the ATP binding site for ATP. Next then we have our thin filaments. We talk about the thin filaments. The thin filaments are twisted double strands of fibrous protein called F-actin or filamentous actin. F-actin or filamentous actin, it consists of many globular, you can see here, many globular actin subunits. So here we've got these globular actin subunits or G-actin subunits. These G-actin subunits, they bear the active sites for myosin heads to attach to during contraction. And you can see those here very nicely. Now in the filamentous actin, you can see they're actually all covered. So Z discs we've talked about. Now let's talk about these actin units. These actin units, these globular actin, G-actin, they're kidney-shaped polypeptide subunits. And they bear active sites to which myosin heads attach during contraction. And then we said that they are covered, and they're covered, as we can see here, by tropomyosin. Tropomyosin is this rod-shaped regulatory protein. It's found spiraling about the actin core. And it covers those active sites. It's controlled by troponin. So the next component you see here is troponin. Troponin is another major protein in the thin filament. It's a globular you can see, three polypeptide complex. One of those polypeptide complexes is inhibitory. It's inhibitory and it binds to actin. Another binds to tropomyosin and helps position it on actin. The third binds to calcium. So they are three globular subunits, and they're written as T, N, I for the first one, the inhibitory, T, N, T for the one that binds to tropomyosin, and then T, N, C for calcium. Each of these, you can see 1, 2, 3 subunits there. So next time when we talk this sliding filament model of contraction, here you can see it states to us basically in the relaxed state, the thick and the thin filaments are going to overlap only slightly. During contraction, myosin heads bind to the actin, they detach, and they bind again to propel the thin filaments towards the M line. As the H zone shortens, the distance between the I bands and the Z discs also shortens. This causes the sarcomere then to shorten. The whole muscle cell shortens, and then the whole muscle shortens. So here you could see exactly that. This is the muscle in the relaxed state. Now after the myosin heads have attached, detached, attached, detached, attached, detached multiple times to allow then the Z discs to move in closer and the H zone to disappear and the I bands to move in closer, here then you have that fully contracted muscle. So also found here you can appreciate, jumping back, is the sarcoplasmic reticulum. Now when we talk about the sarcoplasmic reticulum, it's an elaborate network of smooth endoplasmic reticulum that's going to be found surrounding each myofibril like our sleeve does right around our arm. Now here when we go through and we talk sarcoplasmic reticulum, it's going to regulate intracellular ionic calcium levels. Here, in relation to the sarcoplasmic reticulum, we can appreciate the terminal cisternae. The terminal cisternae are basically the end sacs. They are the end sacs. They are large, perpendicular cross channels that are going to be found at the AI band junction. Next in here we can appreciate running in between two of those terminal cisternae are our T-tubules. Now when we talk about our T-tubules, the T-tubules are going to be elongated tubes. They are elongated tubes that are going to be found at each AI band junction. And what these T-tubules are is they are deep protrusions of the sarcolemma of the muscle cell. So the sarcolemma, the plasma membrane we described it as, it invaginates, it dips down into the cell and comes back out. And why? You're wondering, why does it need it to dip down? Well, if you recall, we said one of the properties was conductivity. It conducts an electrical impulse. Well, that electrical impulse needs to make its way to the center of the cell, to the inside of the cell. Well, why, you must ask? Well, because there's calcium here inside of what you see on the sides of that T tubule that needs to be released. Well, why? Well, because calcium is going to have to come in and bind to the TNC receptor on troponin. Why? So that way it can move the tropomyosin off. Why? So the myosin head can come in and attach. I hope all that makes sense. Beautiful, 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 beautiful. Okay, so we've got calcium. This is all calcium stores. Terminal cistern, where you store. So here we said the sarcoplasmic reticulum is going to be regulating the ionic concentration of calcium. Well, this is where it's going to get released from. Yeah, your job is going to be to find out the name of the receptors. There's two sets of receptors here. You have to know both of those receptor names. First, the electrical receptors that are found inside the T tubule. Okay, well, I'll tell you the names. You're going to have to go through then and find out which one is which. One type are called ryanodine. The other are called DHP receptors, dihydropyridine receptors. Which is which? That's your job to know that. So next then let's talk how the three of these structures, two terminal cisternae and one T tubule, come together to form what we call a triad. So when we talk triad, you can see it's constructed by the two terminal cisternae and one T tubule. And this triad is very important for contraction. So the electrical impulse travels down the sarcolemma, and on the sides, basically you have all these receptors. So voltage changes, those voltage sensitive receptors. And when they start to basically change shape, they are normally in the resting position pushing and blocking the other receptors that are underneath from releasing calcium, because they're the deeper ones of the terminal cisternae. So as soon as these voltage ones get excited, they basically excite the next receptors, which then release calcium, which will flood the cell. Then you know where it goes, and we'll see everything that happens. So let's talk then excitation, contraction, coupling. Excitation, contraction, coupling. Now we talk excitation, contraction, coupling. Here first, excitation has to occur. That's going to happen thanks to the nerve. The nerve is going to get excited, and when the nerve gets excited, it's going to release acetylcholine. Acetylcholine, you have to know, is the neurotransmitter that's going to come in. It's going to bind to these receptors on the postsynaptic membrane. Acetylcholine is going to get released, and it's going to come and bind to the postsynaptic neurons membrane, the receptors that are on the membrane. Binds to those receptors. When it binds to the receptors, it allows sodium and potassium to cross, which then changes the voltage in that area, which then triggers the opening of these voltage-gated channels here on the sarcolemma, which then allow for that action potential to travel across that sarcolemma as we described. So the muscle fiber has to be activated, and then we have to be able to generate an electrical current, the action potential. So here then we can see you've got your two sets of receptors I told you to know. Find out which one is which. I gave you the names. You could do that last part. Next, then we have the propagation of the action potential along that sarcolemma. Right? If we can't propagate that action potential, it's not going to do any good. It's not going to make its way down here. So once it makes its way down here, it's going to be able to release calcium. So the fourth step is releasing calcium. And the calcium then, we already know now what that's going to do. So in order now, so here you can see everything. Now, in order for contraction to occur, you have to have ATP. Remember, ATP has to be on that ATP binding site. If it's not on the ATP binding site, then contraction is not going to occur. So here it's occurring because ATP is on the binding site, so it allows that contraction to occur. And now we also need ATP for detachment too. So ATP for attachment we can see we had. We need ATP for detachment as well. If there's no ATP for detachment, which happens when we die, right? This is all in the part of decomposition. Then we suffer from rigor mortis. Rigor mortis happens again after death, about 1 to 4 days it usually lasts. And then obviously the body starts to decompose more, and then full-on decomposition occurs. Next then is myasthenia gravis I'd like you to know about as well. Now here, myasthenia gravis is a condition where your immune system produces antibodies that block or destroy these acetylcholine receptors on the sarcolemma. Another type is going to block the function of a protein, muscle-specific receptor called tyrosine kinase. So two forms of myasthenia gravis. Let's talk smooth muscle. Smooth muscle. So we talk smooth muscle here. Now, okay, before we jump to smooth muscle, I want you to know what you could do is you could also, we'll Google that video in a second here. So let's talk smooth muscle. When we talk smooth muscle now, smooth muscle is going to be a little bit different here. Now, smooth muscle, here contraction also results from the cyclic interaction of myosin and actin, just like we saw in skeletal muscle. However, the way it's found is completely different from what we saw in skeletal muscle. Here, smooth muscle is going to be found distributed as two layers. You have a longitudinal layer and then you have a circular layer. The longitudinal layer is the outer layer, usually, not always. And these fibers are going to run parallel to the long axis of the organ. Now, when the longitudinal layer contracts, it makes the organ shorter and thicker. It makes the organ shorter and thicker. Next then is the circular layer. When we talk circular layer, the circular layer is the inner layer, usually, again. And the fibers are going to run around the circumference of the organ. When it contracts, it propels contents forward. And when they're doing their job, we talk peristalsis. Peristalsis is a propulsive action. It's where you have alternating contractions and relaxations of basically these muscular layers you have there to help propel food forward throughout this digestive tract. So add in their muscular dystrophy as well. Defective gene for dystrophin causes skeletal muscle degeneration. It's horrible, horrible. All of any disease is horrible. Any of them, all of them are. So let me show you a video here. What you guys can do is go on to YouTube. And when you go onto YouTube, all you have to do is type actin myosin, and we can watch this little short video here that'll depict to us how that skeletal muscle is going to contract. And we're able to watch this little short video here.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
So there you could see then everything animated basically that we went through.